Stem cells, 2009. 98, you remember the first advent of human embryonic stem cells. You have heard about adult stem cells till you're blue in the face, no doubt. And you've probably heard this endless battle between embryonic stem cells will do it all and we don't need anything else. Adult stem cells will do it all. Stem cells from fat will cure the sick. Stem cells from amniotic fluid, cord blood, baby teeth, bone marrow, brains, liver, you name it, it seems to have a stem cell in it. What is the poor lay person to do? So let's cut right to the chase, and I'll give you a fairly simple and I think scientifically robust and accurate way of looking at this. You probably remember early in development, there's that thing called the blastocyst, and inside the blastocyst are a bunch of cells whose job is eventually to make the entire organism or human. Those cells are truly what we call pluripotent. They have the capacity to make every cell type in the adult, and of course pluripotent just means many potencies, right? Pluri is plural, potent is potential. When you look in just about any fetal tissue or adult tissue, you will find stem cells. And the normal job of those stem cells is to keep that tissue going throughout your life. So for example, you've probably heard that 95% of household dust, unless you have a dog, comes from human skin that gets shed throughout the course of the day. Of course, if you have a dog, it's 95% of the dust is dog hair and mud. But I see there's no dog in this house, so it's probably human skin by and large. And of course, if you're losing all that skin, you might ask the question, how come you don't run out? And all that stuff leak out, right? So the answer is you have stem cells in your skin that continuously throughout your life make new skin. Same in the intestine, same in the lung, same in a bunch of other tissues. There are stem cells in the brain. They seem to make new neurons in parts of the brain involved in memory and smell, interestingly. And most adult and fetal tissues appear to have these cells. Now, there's an important point about them, and that is, by and large, actually mostly in the normal course of events, they only make the cells of the tissue they're in. This, of course, makes good biological sense. Imagine that one day you wake up and your stem cells in your brain decided to make liver. This would not be a good way to start your day. And so biology is pretty smart about this stuff. And so they're ordinarily under very tight control. And in fact, stem cells from adult and fetal tissues generally don't make anything outside of that tissue. A lot of scientific papers to the contrary. Generally, the newspaper picks up the part where somebody announces they found something new. They don't necessarily announce the part where we discover that the experiments weren't as strong as everybody thought they were, and we debunk them. So that's the current state of affairs. Cord blood, which you're probably also very interested in, those are blood-forming stem cells. They're basically the same as the stem cells in your bone marrow, wonderful for treating diseases of the blood like leukemias and anemias and thalassemias and things like that. But, turns out, it takes multiple cords to treat an adult. You can't actually grow those stem cells in the lab. Nobody's figured out how to do it yet in spite of over a decade of trying. It's not that the problem won't be solved, it's not solved now. Amniotic fluid, those are like the stem cells you find also in bone marrow that make tendon and cartilage and things like that. Again, very useful, but not the same as an embryonic stem cell. The most recent player, if you will, are these so-called reprogrammed stem cells that truly are something never before seen, where a fellow named Shin Yamanaka in Japan figured out a little cocktail of viruses carrying some genes that normally tell cells how to become pluripotent. And if you put those in a skin cell, you can generate an embryonic stem cell-like cell. A lot of interest in these. They are clearly, as we've started to work with them a lot more, not identical to embryonic stem cells. They're not ready for clinical prime time, but they're enormously powerful agents for discovery. And if I remember, I'll tell you about how we're re-engineering Craig Venter using these in a few minutes. Very interesting topic. Craig just wants new neurons, but we want to redesign him. So that's sort of the general playing field at this point in time. So if anybody tells you there are cells that'll take the place of any other kind of cell, don't believe them. It's not true. Oh, yeah, by the way, those adult stem cells, they're wonderful but they're sometimes hard to get your hands on. So, you know, like those brain stem cells. Where am I going to get those? Tracy? Or maybe I want some from the intestine. Marisa? Just line them up, you know? So the problem is you can't just get these things very easily in the normal course of events without surgery. And that's kind of a problem if you're a research scientist. Okay, Alzheimer's disease. So the statistics on this disease are beyond terrifying, if you know them. So 10% of people over 65 have the disorder. 50% of people over the age of 85 probably have it. I personally am planning on living to be 85, which means that if I don't figure something out, I got a coin flip shot at getting this disease. And it's pretty ugly if you've ever dealt with it with a family member. People lose their memory and their ability to think clearly, and eventually other parts of the brain fail. And it's a pretty long, slow, ugly decline. And it's probably harder on the families than it is on the people who have it in many cases. The problem at the moment is that we really don't have any drugs that do anything very useful. There are a couple drugs like Aricept that give some mild symptomatic relief, but there is no drug that changes the clinical course. There's no drug that slows it down. Once you're diagnosed, you will continue to progress until it kills you or you die of the complications. It's that bad. If we don't find some way of treating this disease, if the current government doesn't bankrupt us, certainly this disease will, because the cost of caring for people with it is just astronomical. And I can say that from firsthand experience. So the question is, what are we going to do about it? Now, the issue is, at the end of the day, we don't really understand what's going on. You know, we're like the 8-year-old auto mechanic who hasn't really figured out how the car works, but we're pretty good at taking it apart, but we don't understand much about what's going on in there or how to fix it. So there are a variety of ideas about the disorder, but at the end of the day, we don't understand why people get it, why some people get it and others don't. What is it that's going wrong in the brain cells? My personal favorite being the movement inside the brain cells, of course. But how, you know, these ideas need to be tested. Now, we do a certain amount of work in mice, and we can take genes that in humans will cause early onset hereditary Alzheimer's disease, which give people Alzheimer's in their 30s or 40s. We can put those in mice, and the mice will develop some symptoms of Alzheimer's disease. But we have a couple problems. The biggest one, of course, is the obvious one. Humans are not just big mice. You've probably noticed this in your daily lives. And that's true in physiology as well. And so the mice get some symptoms of Alzheimer's disease, but they don't get full-blown Alzheimer's disease. And of course, if you think about it, there are certain cognitive tests you might do on human patients